My name is Tamieka Pearce and I'm a PhD student in plant pathology and fungal genetics at the Tasmanian Institute of Agriculture at UTAS. My project is looking at a disease called tan spot in Pyrethrum. Tan spot is a disease that occurs throughout winter and spring in Pyrethrum. It causes really dark necrotic spots and can result in plant death and can wipe out entire fields if not controlled. Pyrethrum is a perennial plant, and within the daisy flower head a chemical called pyrethrum is produced, and that chemical has natural insecticidal properties. They extract that chemical and it is then formulated into a natural insecticide that is used in households and in commercial industries. The incidence of the disease has increased quite dramatically in the last couple of years, so understanding about the pathogen will allow us to control it more effectively. We go out into the field and we collect plant samples that we then bring back to the lab and we isolate the fungal species from them. We then do DNA extractions to extract the DNA from the fungal pathogen, and then we do work using PCR and molecular tools to do work on the DNA. We also have the capacity here at the Tamieka Pearce campus to use greenhouses to allow us to grow pyrethrum all year round so that we can do inoculation studies and see how infection occurs within the plant. When we do field sampling, we take a small piece of the plant tissue that contains the fungal pathogen and put it onto a media that allows the fungi to grow out and then be identified. So for instance, the pathogen I'm working on produces these really cute little curly hyphae that allow it to be easily identified. I'm currently now halfway through my PhD. The work we've done so far has indicated that we have quite genetically diverse pathogen, and that we've also identified 2 different mating types that might allow us to look at how sexual reproduction is occurring. We've also been able to produce a PCR mechanism that allows us to detect pathogen DNA within plant tissue, and hopefully we'll be able to use this to look at how the pathogen moves through the plant once infection occurs. Once my PhD is finished, hopefully we will have enough information about the pathogen that we can create more economically and integrated management approaches to control the pathogen within the field. This will mean that hopefully the cost of controlling the disease within the field will be lower, which will mean that growing pyrethrum will become more economical for farmers, and in turn that also has a better environmental aspect because we will not have to apply as many fungicides to our fields to control the disease. I'm originally from the northwest coast of Tassie, so I went to Hobart to do my undergrad and my honours, but I was fortunate enough that I could travel back up to the northwest and do my PhD at the Tamieka Pearce campus. I could go anywhere in the world and study plant pathology, but I'd love to stay here at UTAS and continue my research. I mean, look around us. Tasmania has got some of the best agricultural facilities and land in Australia, so why wouldn't you want to stay here and study?